Hello dear students, this is Dr. Shabana Begum, Associate Professor in the Department of Zoology, Maharani Science College for Women, Bangalore, presenting a talk on the topic Concept of Serial Homology pertaining to Zoology second semester under the paper entitled Non-Chordata Part 2, Unit 1, Arthropoda and Subunit 1.3, the prawn. Fine. So in my previous session, I gave you a detailed account on the structure of typical appendage, that is biramous appendage, and the structure and functions of various appendages in prawn. And this topic is somewhat related to it. So the content of this topic is the structure of typical appendage and the concept of serial homology with regard to appendages of prawn. Obviously, the learning objective will be the students at the end of this lesson will be able to understand the concept of serial homology in appendages of prawn. So this is the morphological structure of Palaeomon. The diagram will give you the divisions of the body, the various appendages which are serially arranged one behind the other from front anterior end to the posterior end of the body. And also the next diagram here will be the appendages chart, which gives the information about the cephalic, thoracic and abdominal appendages very clearly. These two slides I had already shown you when I explained to you about the morphological structure and the structure and functions of various appendages in Palaeomon. And once again, I am going to make you understand the concept. I am including the same in the particular presentation. So, before starting the explanation of the concept, let me define the term concept. What is meant by concept? So, concept means an abstract idea or a generalized idea, a notion or a conception. Fine. So, here you can mention one more term, homology. Homology means similarity or resemblance of parts or structures in different animals or same animal, and the homologous organs as such are defined as the organs of different animals that are having similar basic structure but different functions. Fine. So here once again, repetition of whatever I just told you. The homology means the similarity due to shared ancestry between a pair of structures in different organisms, while homologous organs have common origin and are built on the same fundamental plan or pattern but perform different functions and have different appearance. Serial homology is the similarity of structure between one part of the animal with another part of the same animal. The concept of serial homology is well illustrated in case of prawn, wherein the 19 pairs of appendages borne by the 19 segments of the body are built upon the same pattern as that of a typical biramous appendage, except the first appendage, that is the antenule, which is uniramus, and all the appendages are serially arranged, that is they show fundamental similarity of structure and modified to perform specific functions. As you all know, a typical biramous appendage, for example a pleopod, consists of a basal protopodite of two podomeres called coxa and basis, and bears two distal rami or branch structures called the inner endopodite and the outer exopodite. Fine. So in the first two slides, I have shown you the diagrammatic representation of Palaeomon as well as the serial, we arrange appendages in case of Palaeomon. And as you all see there and here, I gave the explanation. That is, the body of prawn is divided into three main parts: the head, thorax and abdomen, where the head and thorax together are covered over by carapace to form a combined structure called cephalothorax. Hence, the body is distinguished into cephalothorax and the abdomen. The cephalic region, that is the head, bears 5 pairs of appendage bearing segments, and the appendages are antennae, mandibles, maxillulae and maxillae, starting from anterior, that is in front, backwards respectively. Here the antennae are tactile, that is they are sensitive to touch, and also they help in balancing of the body because of the presence of the balancing organ called statocyst, which is present in a small concavity of the coxa of this appendage. The antennae are also tactile and excretory in function because of the presence of excretory pore in the coxa of this antenna. The mandibles are masticatory, that is they help in chewing the food, while the maxillulae help in food manipulation, while the maxillae help in food manipulation as well as respiration. Next, the thorax is 8 segmented and bear 8 pairs of appendages. That is, the first 3 pairs are called maxillipedes and the rest 5 are called the walking legs, also called as periopods. The maxillipedes help in feeding and holding the food in position, while the mandibles masticate it. The walking legs are of 2 types, with chelate type, that there may be 2 or 3 depending on the type of prawn. They help in holding the food and passing it into the mouth, and also serve as organs of offense and defense, while the non-chelate legs help in walking. Finally, the abdomen is 6 segmented and bears 5 pairs of pleopods, also called swimmerets, helping in forward swimming and also serve to carry eggs, while the 6th that is the segment, bear a pair of uropods. They are the appendages of 6th segment called uropods, which along with telson, which is a conical structure, the last point or last part of the body of the prawn, helps in backward swimming movement. Fine. So, from the above description, it is very clear that the basic structure of all the appendages in case of prawn is similar or same, but their function is different. Hence, one can conclude that the concept of serial homology is well illustrated in case of prawn. Fine. With this, I will stop this small topic, and you may get 3 mark or 5 mark questions out of it. So you just go through the slides once again. Fine. Thank you.